So it's what I would call an adulteration because it wasn't disclosed, and it means it's an added thing that was put in that was not on the label. And for everyone around, the reason why the FDA and Health Canada was first made an agency was to regulate adulteration, mislabeling, and contamination back in the early 1900s.
Tamara Ugolini here with Rebel News to bring you more reports on Plasmidgate, this time from Maria Gucci, who is a pharmacist by training with some experience in infectious diseases. Gucci spent over a decade working in a regulatory environment where her primary responsibility was to regulate drug pricing in Canada. Through that work, she learned how the pharmaceutical companies market and price their drugs, including how they use scientific information in order to achieve their end goals. This garnered Gucci a role as a scientific advisor where she gained insights into how to assess drugs from a comparative point of view and dig into both clinical and non-clinical trials to compare those drugs to determine their best pricing. As the novel COVID-19 mRNA injections became authorized in Canada under an interim order in December of 2020, Gucci began digging into the European Medical Association's medical report submitted by the manufacturers Pfizer and Moderna, a system that she was familiar with from her past work, and she started to notice massive discrepancies between what the pharma manufacturers were submitting and what the regulators were asking of them, at which point she concluded that these products did not meet good manufacturing processes or basic regulatory standards.
Do you want to start feeling like your pre-COVID self again? You're not alone. The wellness company's Spike Support Formula is an all-natural supplement to help people do just that. It was created by cardiologist Peter McCullough and his expert team of doctors to help the people experiencing effects from COVID and the you-know-what.
Go to twc.health/rebel today.
That was a point of discussion when we first interviewed Gucci in November of 2021, at a time when she was joining us anonymously. Well, today she has an update after the recent publication of her co-authored paper titled DNA Fragments Detected in Monovalent and Bivalent Pfizer/BioNTech and Moderna Mod RNA COVID-19 vaccines from Ontario, Canada, exploratory dose-response relationship with serious adverse events. Gucci is going to discuss the multifaceted factors at play here, many of which compound one another. It all starts with how the manufacturers of these novel injections changed their manufacturing process from what was used in the clinical trials to something different, a change in the manufacturing process that allowed the manufacturers of these novel products to essentially warp speed the injections into the arms of billions of people worldwide. Check it out.
That in the clinical trials, what was done was a qPCR kind of thing where you basically it is genetic engineering that makes the DNA, and from that DNA you make the mRNA. But that can't be scaled up very fast because it's slow, and it's a slower process and takes a fair amount of time. So plasmids put into E. coli is easier, faster, and because E. coli replicates in 20 minutes, so you can get a new plasmid in 20 minutes. So you can make a lot in a short period of time, and so that was a natural progression. What happened, though, is as they made the mRNA from a plasmid-based system rather than previously, what happened is that they had a lot of problems purifying it and making it comparable to the original that was done in the clinical trials. And from a regulatory perspective, then, do we know that this new product is going to work as well as the old one? So that was the big issue. So we have no comparison directly clinically, whether you're going to get the same clinical outcomes or based on the product that was had decreased amount of mRNA, actually, active mRNA. That was the major concern from it. In addition, it had a bunch of other impurities in there that wasn't in the process 2, that wasn't in process 1, but that was inside process 2. So that was a very different product. Doesn't necessarily mean it can't be used. It just needed to be compared clinically. And that was part of the problem. That has not been done, or we're not sure it has been done in the manner that you would normally expect in a normal regulatory kind of process for biologics. So we have many issues here, many issues. So the comparability between process 1 and process 2, I think the regulators are going to say, we've had enough experience in the patient population. Basically, they were the experiments, and that they're considered comparable. On top of that, you have your bivalents and your children's vaccines that were based on process 2, and process 2 not really compared to process 1. So in my opinion, it's sort of like everything is extrapolating on top of each other so that you have nothing anchoring you to the actual clinical trial. Having said that, a lot of people would say the clinical trial isn't the best either. That's true. But you can't anchor everything back to the original clinical trial that you would for normal drugs and biologics. So it's one on top of the other on top of the other, and they're all an extrapolation based on the previous. So it gets more and more nebulous, I would say. So that's problem number 1. So the other thing, the other regulatory aspect that is of concern to me and several people is that we really have, part of the problem is we really have a gene therapy product. It's not gene therapy as defined by many people, but it's a gene therapy product because you're transfecting, you're bringing a nucleic acid from outside the body into the cytosol. That's transfection. Usually there's different ways of doing it. Lipid nanoparticles is one. You can actually use a gun, a DNA gun that pushes it in. But what you're doing is transfecting. And when you do that, that is a genetic product because RNA is still, nucleic acid is still genetic material. And when you do that, we're not thinking like a regular vaccine anymore. We're thinking, and because of that, some of the non-clinical trials and assessments, especially for long-term adverse events, is not done for vaccines and should be for gene therapy type products. And that is my feeling, or actually I feel quite strongly about this, that these products should have been assessed as a gene therapy product. So that's another regulatory issue that needs to be addressed. And then finally in our paper, because of process 2 being one based on plasmids, and this is normal, this is not the issue, but the issue here is that we have found impurities of DNA. And one of the big issues again is, and people don't realize this, we have this whole new platform. How do you test the purity? How do you know what is pure if you've never done it before? So you have a bunch of analytical methods that you don't know is appropriate for this or not. So the analytical method for measuring the DNA leftover, which is leftover from making the mRNA, and that's normal. We want to control that impurity to as little as possible, okay? The issue that we have is that our my collaborators have determined that based on qPCR, which is the test that's used by the regulators, they all meet the criteria that was set down for vaccines, okay? But if you use another test, the qubit, which measures small pieces of DNA, less than 200 base pairs, there's quite a bit of that in there. So what does that mean when you transfect all these small pieces of DNA into the cell? Is this a problem or not? We believe it is. And that's a discussion to be had and to be looked at. And we feel that that needs to be looked at. And then lastly is the discovery of the SV40, which is a very serious thing because it was not disclosed to Health Canada as being as part of the plasmid. And this, in my view, is extremely serious because it is a requirement under the Food and Drug Act that manufacturers or anyone making a drug disclose the material parts of the product that you are authorizing. So it's like I have a structure of a new drug and it has a big side chain over here, and I don't tell them there's this side chain. So that is something that's what is missing. So it also is an addition. So it's what I would call an adulteration because it wasn't disclosed, and it means it's an added thing that was put in that was not on the label. And for everyone around, the reason why the FDA and Health Canada was first made an agency was to regulate adulteration, mislabeling, and contamination back in the early 1900s. Do you remember the term snake oil, right? And that's because people, manufacturers of liquids at that time, and cough syrups would put morphine in them, not tell the parents, and say, oh, this will stop the cough in your kid. Well, sure it did. It stopped everything. So this was undeclared ingredients in this. That's called adulteration. It's in the act, and it's really considered quite a serious issue. Having said that, it's up to Health Canada to decide whether or not it's serious enough to put any kind of, what's the word, whether to remove it from the market or whether to fine Pfizer or any kind of remedies they want to make. That's up to Health Canada to decide. But the SV40, just the fact that it's there, even if it didn't do anything necessarily, is an adulteration. The second part of that is that the SV40, other people can explain this much better, but it has been used for gene therapy experiments. It is known to cross into the nucleus. In addition, it's small. It's only 72 base pairs. So if you're doing qPCR, you're going to miss the SV40. That's why we think the other qubit thing that measures pieces of DNA under 200 is means that it's getting weird. Since we measured it, it means the SV40 could be part of that in the cell.
Right. Now, I mean, the SV40 point in and of itself should be enough for Health Canada to pull the brakes on this and say, whoa, we need to maybe take a temporary pull off the market, review things, do some more investigations, and determine whether or not these products are as safe and effective as we've been touting for the last almost 3 years.
Yeah.
Instead, they haven't done that. They've really bogged down. They're clinging onto that safe and effective narrative. This coupled with now the undisclosed DNA fragments, this seems to me like a giant mess. How do we get out of here?
That's a very good question. It is a giant mess. And it, I just don't want to leave Moderna off the hook here, okay? Because Moderna does have quite a bit of small fragments in there. They got rid of the big ones, which you see on qPCR, but they have quite a bit of small ones. So they just don't have the SV40 that we can determine. So they are both have a lot of impurities of leftover DNA in there that we feel at the very minimum that there are other methods and techniques to get rid of that should have been that the regulatory, what we call compendial standards need to be set higher, better, and reviewed so that this does not continue on. At the very minimum, we also need to determine if this is a risk for patients or is it not a risk. We don't know.
In closing, Gucci calls for a review on how these products were reviewed under that interim order, otherwise known as emergency use authorization, including how they were reviewed under the rolling submission process, which meant that the manufacturer could literally submit safety data on a rolling basis as it rolled in, which means that if the data showed that there were safety or toxicity issues, it would be hard for both the regulator and the manufacturer to backpedal on those safety assertions as the product was simultaneously being and had been injected into the arms of millions and then, of course, billions of people. Especially as evidence of objective and measurable DNA contamination risks mount, leading to things like myocarditis, clotting, and neurological effects, among other unknown adverse events, many more are now wondering, do the once purported benefits of these products really outweigh the potential risks? I've reached out to Health Canada to inquire just that, so please stay tuned at nomoreshots.ca. For Rebel News, I'm Tamara Ugolini. At that website, nomoreshots.ca, you can sign our petition and also send an email to the federal Minister of Health, that's Mark Holland, calling on him to revoke Health Canada's authorization of these novel mRNA injections. You can also chip in there if you're able to, a few dollars, to show that you support these journalistic efforts and this work on this topic. Exercise your democratic voice and let us know that you appreciate reports just like this at nomoreshots.ca.